We now have even stronger evidence that the ME/CFS brain, or the myalgic encephalomyelitis chronic fatigue syndrome brain, has abnormally increased lactate. And this indicates a neuroinflammatory issue, abnormal activation of the microglia in the brain, oxidative stress, or maybe a problem with basic energy or some mitochondrial issue. Now, if you follow my videos, you know I've seen increased lactate in the ME/CFS brain since the first paper we put out in 2020. And now there's even more support for this observation by other groups. And I can tell you as a scientist, one of the best things for us is when our observations are supported by the work from an independent group that's using different techniques, and everything just kind of fits together, and it lets us know that we're really on the right track. So there's this exciting new paper. This group looked at brain lactate in ME/CFS and in long COVID. And you can check out my earlier videos about why increased lactate is a marker of brain inflammation. I'll put a link to one of the videos at the end of this one. But this group used magnetic resonance spectroscopy, just like I do, but they used a 7 Tesla MRI scanner. And almost all research on brain scans are done on 3 Tesla scanners, including most of the work that I do. So let me show you the difference between a 3 Tesla scanner and a 7 Tesla scanner image. And these are images from the Mayo Clinic. And this is a top-down view of the brain. We just call this a standard structural image. And at standard 3 Tesla, you can see these major brain structures, and that gives us a lot of great information. But at the much more powerful 7 Tesla magnet, you have greater clarity of the smaller aspects of the cortex. You can really, without even me describing it, you can see the difference here. It increases what we call signal to noise ratio, and that gives us a lot of good things. But one of those is to increase the resolution of the images, so we can zoom in and really find things that might have been missed at 3 Tesla. So I really like when people use 7 Tesla. They're more complicated to use because the magnet's stronger, and that introduces a host of issues. But when it's done well, we get some really good information from it. So anyway, the new paper is Brain and Muscle Chemistry in Myalgic Encephalomyelitis, Chronic Fatigue Syndrome, or ME/CFS, and Long COVID, a 7 Tesla magnetic resonance spectroscopy study. And this is an open access paper. So you have the same access that I do, and I put a link in the video description if you want to read the paper yourself. Now, they looked at 24 people with ME/CFS, 25 people with long COVID, and 24 healthy controls. And these are very nice, equally sized groups, which you don't always see in papers. Sometimes they have a whole lot of healthy controls, but only a few patients. And then you're always a little suspicious that that could affect the statistics. But this one, there's really nice equal sample sizes per group. Now, they did a few different things, but I'm only going to talk about the brain lactate today. What they found in long COVID is also interesting, but I'll probably talk about that at a later time because it's important for different reasons. So here's a side view of the brain to show where they imaged. And they imaged 2 regions. One is called the pregenual anterior cingulate cortex, that's in green. And the other one is the dorsal anterior cingulate cortex, which you can see in blue. And again, if you follow my videos, you know that these are very important regions in the type of work that I do. And I think they're very important in ME/CFS. So I think the regions they selected was great. I think they picked really good regions to look at. Now, their approach requires them to select specific regions to assess. And so their approach doesn't cover as much of the brain as the approach that I use, which covers the whole brain, but they have greater sensitivity to find differences. So there's pros and cons to each approach. You can image more stuff, but with lower sensitivity, or you can focus in a region. You may be missing things in other parts of the brain, but you have a really good sensitivity to find things in that region you're looking at. So it really just depends on your question, which one makes more sense. All right, so this is what the scan provides. It gives us concentrations of different chemicals in the brain, and they're measured by the height of the peaks. And because this group is using a 7 Tesla scanner, they're able to separate out more chemicals to measure than we might be able to do with 3 Tesla. Anyway, the one that we're interested here is the lactate, which is right here. I just put it in this tiny little green circle. Now, this is a small peak relative to many of the other things you can see on this plot, but lactate should never be super high. If your lactate was much higher than what you see here, that would suggest potentially a medical emergency. And in normal cases, this peak shouldn't be visible at all. So, and that's because you really, in normal circumstances, you really shouldn't have measurable lactate in your brain. So any peak is interesting. All right, so I mentioned they recruited 24 patients. Now they used the CDC FACUDA criteria for Centers for Disease Control, FACUDA. Now this is a fairly lax case definition compared to other definitions. So it's the easiest definition to meet. For example, it doesn't require the presence of post-exertional malaise or PM, and that's something I'll talk about in another video. Demographically, the groups were pretty equal, except for the ME/CFS group was significantly younger than the long COVID or the healthy groups. The ME/CFS people averaged in their early 30s, while the 2 other groups were in their early to mid 40s. Now, I can't think of any reason off the top of my head for the age difference to be the cause of any lactate difference. But of course, it's always better if there's no difference in age between groups, just to rule out that possibility. So it's just something to keep in mind. All right, so they did the magnetic resonance spectroscopy scan, and all the technical details are in the paper if you want to see those. And getting right to the main finding, they found that the lactate was elevated in the ME/CFS group in both the pregenual and dorsal anterior cingulate cortex. Those are both regions that they tested. So what they were, I think, expecting to find, they found. Now, these elevations that I'm showing right here, they don't look extreme, but as I said before, even relatively small increases of lactate can reflect significant pathology. So that's really the main story. Again, there's other things I could talk about, but I like to keep these pretty focused so they don't get too confusing. Lactate elevations in both of these regions could drive what we call the sickness response, which is profound fatigue, maybe pain sensitivity, depressed mood, feelings of social isolation, decreased motivation, and anhedonia, so a decrease of pleasure. And really, the shortcut to thinking of what this is like is all the stuff that you feel when you have a horrible flu. That's what these regions control, or potentially when you have ME/CFS. Those are the types of symptoms we're talking about that we generally call the sickness response. It's our brain's response to illness. So what do we do with this? Well, these findings are really interesting. It supports our hypothesis that we've been working on for a long time here, and it does suggest a few targets for therapeutic compounds to be tested. If you follow this channel, you know I've been pushing hard for clinical trials that specifically target inflammation in the brain, and I will have important updates on that topic over the next few months. I know that what ultimately everyone wants to hear is what treatment can fix the problem. We cannot definitively answer that question right now, but we're moving in that direction. And studies like this do help guide me in deciding what treatments to test. And so I do think that these imaging papers are still very, very important. But as much as I love neuroimaging, and I've been doing neuroimaging for probably 20 years, I have been shifting as much of my effort to clinical trials as much as possible so we can get treatments out to clinicians and patients. We've had decades of compelling mechanistic findings in ME/CFS and related diseases, and that work should definitely continue. But it's past time for us to just take everything we know, prioritize the best potential treatments, and then put them to the test. And I'm far from the only one who believes that. I think most scientists in this field would agree with that. So do follow the channel if you don't already, and I will keep you up to date on all the newest scientific findings. That's what I do when I'm not writing. I'm reading the new papers that come out this week, so I can stay on top of the field and I can catch things when important things come out. Especially for this channel, if a study comes out that indicates a new potential treatment, I will be sure to prioritize that. So I do thank you for listening. I hope that's helpful, and I will be back soon.